We are running out of fuel.
There's not much time either. With the Earth's fossil fuel reserves depleting, we are shifting our focus to sustainable solutions for powering our world.
Within the field of green energy, research is constantly evolving to broaden our understanding of biofuels and their practical applications. One of the most used resources to produce bioethanol is corn. However, corn has its downsides. It needs a lot of land to grow, and the process of converting corn to biofuel requires more resources than it produces.
There exists an ancient bacterium that has terraformed the Earth once already. Theorized to be the world's first oxygen supplier, this marvelous little microorganism changed the world. Kind of like Gandalf. Cyanobacteria, also known as blue-green algae, has the potential to revolutionize the world once again.
This time, instead of oxygen, it can provide us with energy.
And how can it do that?
Well, the strengths of cyanobacteria are many. They grow fast, they only need sunlight to absorb carbon, and can grow in seawater. They're perfect for candidates for sustainable energy production. This is how we can use cyanobacteria, specifically bacteria PCC7942, to produce bioethanol.
Just don't drink it. But PCC7942 does not produce ethanol on its own. It needs some help. Gandalf needs assistance.
Researchers were smart enough to genetically insert DNA from a different species of bacteria, Zimomonas mobilis, inside our cyanobacteria.
What?
We can just call him Zimon. By borrowing some of Zimon's DNA, our bacteria can now produce their own ethanol.
Hold on, how can they do that exactly?
By taking a plasmid which contains genetic material from Zimon, we identify the two regions we're interested in. We then place them into our cyanobacteria. The regions are cut by a restriction enzyme. Thanks to a shuttle vector, we can insert the cut regions in the bacteria. Our bacteria is now ready to produce some vodka.
What?
Ethanol. I meant ethanol.
On its own, our bacteria is only able to produce pyruvate. This process is a dark reaction. It doesn't need sunlight. With the help of our brand new enzymes, we can now convert this pyruvate to ethanol. Pyruvate decarboxylase, also known as PDC, converts pyruvate into acetaldehyde. Let's look at this interaction in action.
Pyruvate is a polar molecule and is kept in place by other amino acids. When the cofactor binds, the PDC undergoes a conformational change and the glu-473 residue comes closer.
This residue undergoes redox reactions with the cofactor. This allows the pyruvate to bind and split in two. Finally, the cofactor is restored to its original state and acetaldehyde is produced. Let's now focus on ADH.
The NADH sticks to the protein through an NADH-specific binding site, like a tiny pocket. At the same time, acetaldehyde is held by nearby polar residues. Iron mediates the process by bringing NADH and His267 together. This catalyzes the transfer of a proton to acetaldehyde. And by the end, this transfer results in the production of ethanol.
Awesome. It is no surprise that PCC7942 is a pretty funky organism.
Through research, we can create a cheaper and more efficient process for producing biofuel from cyanobacteria. It's time to begin the shift to sustainable energy solutions.
A special thanks to Zimon and Gandalf for sponsoring this reaction. Ciao, bacteria.